Mouse tryptase gene expression is coordinately regulated by GATA1 and GATA2 in bone marrow-derived mast cells. Now you might be asking, what the hell does any of that mean? Don't worry, I'll tell you. Let's start from the beginning. If you have any allergies, you know what it's like to have an allergic reaction. Puffy eyes, red itchy skin, an upset stomach. Maybe you even have trouble breathing or feel like passing out. Some people's allergies are milder, and they can just take some over-the-counter medication like Benadryl to relieve the symptoms, but some people aren't as lucky, and they can go into anaphylactic shock. Anaphylaxis is a severe, life-threatening reaction. We know what it looks like on the outside of your body, but what about the inside? Anaphylaxis is caused by the activation of mast cells. When mast cells are activated by a trigger, the allergen, they release compounds called mediators that cause reactions leading to symptoms. An important one is tryptase, which plays a crucial role in mast cells' function and inflammation. In mice, a lack of mast cell tryptases results in mice being more susceptible to infection by bacteria and parasites, which shows us that these mast cell tryptases are important in the immune system as well. The genes that encode mast cell tryptases are on different chromosomes in humans and mice. The tryptase locus in mice encodes a couple of different genes expressed in mast cells, but for this study, we're focusing on TPSB2 and TPSG1. As we know, gene expression can be regulated by transcription factors, as they can bind to DNA and either activate or repress their expression. Essentially, they can turn them on or off. As far as the mast cell tryptase genes go, studies have shown that MITF is involved in their regulation, as are GATA1 and GATA2. While the researchers were aware that these transcription factors were involved in the regulation of mast cell tryptase genes, the exact mechanism behind their function was unknown. Therefore, the researchers decided to conduct a study to investigate how GATA1 and GATA2 specifically regulate tryptase gene expression in mice mast cells, more specifically, bone marrow-derived mast cells, BMMCs. Well, how did they go about doing this? First, the researchers needed cells. The researchers first received mice with conditional knockouts of the GATA1 and GATA2 transcription factors, and then they extracted bone marrow-derived mast cells from the femurs and tibia of the mice. Next, they cultured these cells in growth medium. The supplements provided in the medium allow cells to properly divide and multiply so that they had enough cells to manipulate. To examine the mechanisms of GATA1 and GATA2 function, the researchers needed to observe the consequences of the loss of expression of these transcription factors. They did this by introducing small interfering RNAs that silence the expression of genes. Think of them as a mute button. siRNAs can do this through a process called RNA interference. As mentioned, MITF is also a regulator of tryptase gene expression. The researchers also wanted to test if MITF expression could rescue tryptase gene expression in the GATA1, GATA2 conditional knockout BMMCs. If it can't, it would show that GATA1 and GATA2 activate tryptase gene expression independent of it. To do this, they took a plasmid they inserted the gene encoding MITF into and transfected the cells with this plasmid so that the gene will be transcribed and provide the cells with functional MITF. Next, the researchers isolated RNA from the BMMCs. From there, they converted the RNA into cDNA, which simply means that the DNA is complementary to the RNA. The cDNA was then used in an RT-qPCR reaction. PCR simply amplifies the amount of transcript you have, so the researchers used it to be able to have a greater amount of cDNA than the limited amount they were able to get from the mice. The Q is for quantitative, which means that this method was used to quantify the amount of tryptase gene expression based on the amount of transcribed RNA they isolated. This is to determine if the silencing of GATA1 and GATA2 with siRNA had an effect on tryptase gene expression. If it did, we know that GATA1 and GATA2 regulate those genes. After this, the researchers used ChIP and ChIP-seq processing to study the interactions between specific proteins, the transcription factors, and DNA. The use of ChIP and ChIP-seq allows for the identification of GATA1, GATA2, and MITF binding sites in the tryptase genes, and it would show the researchers where exactly in the genome the transcription factors bind to tryptase genes. Lastly, the researchers used CRISPR-Cas9 genome editing. This allowed the researchers to delete the region on the tryptase genes that bind GATA1 and GATA2 to determine the consequences in altered expression, if any. After analyzing their results, what did they find? First, they discovered that the introduction of siRNA targeting either GATA1 or GATA2 into BMMCs leads to a significant reduction in mast cell tryptase gene expression. With analysis of RT-qPCR results, the researchers found that 24 hours after the introduction of GATA1 and GATA2 siRNAs into the BMMCs, both tryptase genes expression was significantly reduced. The researchers also decided to look at the effect of GATA1 and GATA2 activity reduction on other tryptase genes to see if perhaps one transcription factor does more than another. This is indeed the case. GATA2 silencing resulted in the reduction of other tryptase genes listed here, but there was not much reduction in the expression of these genes with GATA1 silencing. That's what this graph here shows. If we draw a line here, we can see how much more mRNA for all of these genes there are in the GATA1 knockouts compared to GATA2. The same thing goes for the two specific genes we're focusing on. In addition, the researchers also looked at RT-qPCR results for the effect of GATA1 and GATA2 silencing on the other transcription factors that can regulate tryptase gene expression, which I mentioned earlier to be MITF. We can see that there is a clear reduction in MITF mRNA in the GATA1 and GATA2 knockout BMMCs. Next, the researchers discovered that the overexpression of MITF failed to restore the reduced TPSB2 transcript level in the GATA1 GATA2 double knockdown BMMCs. Since the researchers discovered a decrease in MITF mRNA in the GATA1 and GATA2 knockdown BMMCs, they thought that maybe the GATA transcription factors indirectly regulate the tryptase genes through MITF. MITF undergoes alternative splicing to produce two different versions, MITF-A and MITF-MC, both of which having their activity reduced by GATA1 and GATA2 silencing. The researchers needed to test if overexpressing MITF could rescue the expression of TPSB2, one of the tryptase genes, in the GATA1 GATA2 knockdown BMMCs. Through analysis of RT-qPCR results once again, it was found that the overexpression of either MITF isoform did not affect the transcript level of the TPSB2 gene in the GATA1 GATA2 double knockdown BMMCs. Therefore, GATA1 and GATA2 regulate tryptase gene expression independently of MITF. Next, the researchers found that GATA1 and GATA2 bind to 3 genomic regions upstream of the TPSB2 gene. The researchers aim to identify specific DNA sequences essential for the regulation of mast cell tryptase genes by GATA1 and GATA2. They used publicly available ChIP-seq data to identify potential regulatory regions, and they discovered several GATA2 binding sites on the tryptase gene locus on the mouse chromosome, specifically the -72.8, -63.4, and -1.1 regions. When analyzing their results, they found that the GATA1 binding sites are the same as GATA2. The 3.9 and 27.7 regions, which are the last two on the graph shown here, are control regions. As we can see, there is a similar binding pattern for GATA1, though GATA1 binding to these regions was weaker than GATA2. The binding of GATA1 and GATA2 to the tryptase locus is the strongest at the -72.8 and -1.1 regions. The results suggest that GATA1 and GATA2 may regulate the expression of the tryptase genes by binding to these regions in the genes. There's one more thing the researchers found that is very important. The researchers discovered that the GATA2 binding activity to the -72.8 region specifically is reduced by GATA1 ablation. The researchers took a closer look at the results to examine whether or not GATA1 and GATA2 affect each other's binding to these regions. Interestingly, GATA2 binding to these regions was significantly reduced in GATA1 knockout BMMCs, indicating that GATA1 is required for the maximal binding activity of GATA2 to these regions. Cooperative binding. The results indicate a complex interplay between GATA1 and GATA2 and regulating their binding activities to specific genomic regions that influence each other's binding, and therefore they collaborate in regulation of mast cell gene expression. Because both GATA1 and GATA2 work together to regulate the expression of tryptase genes, we can conclude that they coordinately regulate the gene's expression. And that's it. Clearly, the researchers did a lot of work and were able to further understand the regulation of tryptase gene expression. However, there are limitations to this study. The study primarily focuses on bone marrow-derived mast cells, BMMCs. Findings from these cells may not fully represent the complexities of mast cell biology in other tissues or contexts. Generalizing the results to mast cells in various physiological conditions or tissues might be limited. Next steps would be to investigate mast cell biology in different tissues and physiological contexts to better understand the diversity of GATA factor roles. In addition, the study assesses gene expression at a specific time point, 24 hours, after siRNA transduction. Mast cell biology is dynamic, and changes in gene expression over time may provide a more comprehensive understanding. Multiple time points could reveal temporal dynamics in the regulatory processes. Next steps would be to conduct time course experiments to capture dynamic changes in gene expression over multiple time points, allowing for a more nuanced understanding of GATA factor-mediated regulatory processes of tryptase gene expression. To restate what we've learned, GATA1 and GATA2 ablation results in decreased expression of the tryptase genes TPSB2 and TPSG1. GATA1 and GATA2 ablation also reduced expression of another transcription factor of these genes, MITF, but GATA1 and GATA2 work independently from MITF to regulate tryptase gene expression. Lastly, GATA1 and GATA2 specifically bind to 2 regions on the tryptase locus, and GATA1 and GATA2 affect each other's binding to these regions. They coordinately regulate tryptase gene expression. Well, why do we even care about any of this? What could we possibly apply these results to, or how could we use these results to help us do other things? First, investigating the regulatory mechanisms of mast cell tryptases can provide insights into the functional role of mast cells in immune responses and inflammatory processes, which can help us understand these processes not only in mice, but in humans too. Next, mast cells and mast cell tryptases play a crucial role in allergic reactions and inflammation, as we noted early on. Therefore, understanding their regulation can lead to the development of targeted therapies for allergies, asthma, and other inflammatory conditions. It may help researchers to identify potential drug targets for stabilizing mast cells, which could be beneficial in preventing excessive immune responses. Lastly, understanding how GATA transcription factors affect mast cell tryptases can also provide insights into disorders and cancers like mastocytosis and certain types of leukemia that are often associated with excessive levels of tryptase. We can see that while this research may seem very specific and not very useful, these applications are clearly very useful in both basic research and clinical settings and give us an appreciation for what the researchers have done in this study.